And this evening, it's a tremendous pleasure to welcome Sir Paul Nurse, President of the Royal Society and Director of the Francis Crick Institute in London, to give his lecture on good ideas, great ideas in biology. Paul is very well known to all of us in terms of his achievements and his science. He is, of course, a winner of the Nobel Prize with Leonard Hartwell and Tim Hunt for their discoveries in the chemistry of protein molecules that control the division of cells. And I just want to orientate you about what we're going to hear this evening. What we're going to hear is about the kind of mechanical and chemical control within cells which gets them to do all the hugely intelligent things which the cells of plants, animals, and so on do. It's a very, very fundamental issue of switching and manipulation and organization. That's the field that Paul is in, and I'm sure he will give us a rivetingly interesting insight into that process. I look forward to it very much, and you will have the opportunity at the end of the lecture to quiz him. We're going to have the lecture and then follow that up with the discussion. As I've said, you will be able to ask the questions which you want to. So that's enough from me. As President, I hand over to the President of the Royal Society. This is the Royal Society of Edinburgh.
Well, thank you, John, for that introduction. It's a real pleasure to be here. Twofold, really. One is to be present at the Sister Academy, the Royal Society of Edinburgh, of which I am an honorary fellow. I'm privileged to be. And secondly, to be back in Edinburgh, where I spent 7 years of my research life in the 1970s. And it's always an absolute delight to come back to Edinburgh and to see, as I have done already today, so many people that I know. I have two slides at the beginning which indicate my jobs. I'm President of the Royal Society, so that's this one. And I'm also setting up an institute next to St Pancras Station and King's Cross called the Francis Crick Institute. So I just thought I would show you a picture of that as well. And that will open at the end of next year. But today I'm going to talk about the great ideas of biology. Sometimes I call it the great ideas, and then I feel nervous about that and get modest, so I call it great ideas rather than the great ideas. And, you know, on the whole, biologists, I'm a biologist, of course, tend not to talk about great ideas and grand theories. In this respect, we're rather unlike the physicists who love doing that sort of thing. We tend to be, tillers of the soil. We're more comfortable with details and particulars. You know, we like to list the numbers of species in a habitat, for example, count the hairs on a beetle's leg if we were a Victorian entomologist, sequence genes if we were a modern molecular biologist, or determine the RNA and proteins in cells. So we don't talk so much about great ideas, but there are some great ideas in biology. I don't think they quite get talked about enough. And I'm going to talk about 4 of these tonight, and I'm going to point in the direction of a 5th, which I personally think is interesting and important, but is yet to be properly formulated. And I hope as a consequence of this that you'll have communicated, I hope to have communicated a sort of better insight into the nature of life, which is really, I think, one of the key interests, of course, of biology. But it's not a particularly technical talk. Indeed, it focuses more on the history of ideas and is even a little philosophical at times and emphasizes connections between different approaches. So it's a sort of, it's a bit of a mix match, and I hope everybody at different sort of interests and levels of biology will get something out of it. So I'm going to start with the first great idea I want to talk about, which is the cell as the basic unit of life. And this idea is very easily stated. It's that all life is composed of cells, so it's the basic structural unit of life. and that the cell is the simplest unit which exhibits the characteristics of life. So that's a more functional statement. In a sense, you could consider the cell as life's atom, the basic unit of life. Now, this idea is actually an old idea. In fact, most of the ideas I'll talk about are quite old. It started in the 17th century, and it really took around 200 years to completely and fully develop. And as is often the case in science, it was technology which begat discovery. And in this case, it was the invention of the microscope, which was invented in the Netherlands in the early 17th century, and was then used in the mid-17th century by Robert Hooke to look at biological material. And you'll see his microscope that he used there on the left, a compound microscope. And what he did was to take a razor and to cut a slice off a piece of cork, actually, and he drew the black and white picture that you see there on the left. And there's a modern scanning EM on the right to show you what that would look like today. He observed all these little boxes, and he called these cells. I've read somewhere, but I can no longer find it, so I hope I just didn't dream it, that he thought this reminded him of a sort of plan of monastic cells in a monastery, and he somehow made that connection. That may be complete hooey, but it might be right. Now, once he had made that description, and I should say something actually before I move on from Robert Hooke, he was the curator of experiments for the Royal Society, and he was like the worker who did the experiments, because the Royal Society was, unlike the Royal Society of Edinburgh, a society of gentlemen who didn't actually do very much. They sort of just got together, and they needed somebody to demonstrate the experiments, and that was Robert Hooke's job. So he was the worker there. And once Robert Hooke, who incidentally fell out with Newton, who probably destroyed most of his images, so we don't even have any pictures of him that are completely clear, because he had a big row with Isaac Newton, who was subsequently a president of the Royal Society and a deeply unpleasant person. But of course we revere him for, laws of motion and gravity and all that sort of thing. Now, once Hooke had seen cells, as is also very often the case in science, everybody else saw them. I mean, once you know what you're looking for, you see them. And within about 15 years, images such as the one there drawn by Nehemiah Grew, who was a physician and a fellow of the Royal Society, of a vine there, fantastically detailed. I mean, it would have been appropriate for a mid-20th century book on plant anatomy, was being drawn. This is a modern picture, again, on the right over here. I actually have the book from which this picture is taken, and it's full of fantastic plates that look like this. So plants were easy to look at, of course, and so it was clear that cells were prevalent in plants. But also at the time, others, particularly Leeuwenhoek, was using, in fact, a much simpler microscope, a single-lens microscope, which was of much higher quality. And he discovered single-celled life, microorganisms. And what he did is he scratched in between his teeth and put the stuff that's in between our teeth on a slide, looked under the slide, and drew these objects, which are bacteria. I particularly like this one doing, you see, he drew it, he must have done a loop-the-loop underneath the, on the slide over here, and so discovered single-celled life. And this was about 1685, 1690, all within a few years of Hooke's original discovery. Now, there's one image of Leeuwenhoek, but there's something unusual about him. He was a draper who lived in Holland, in Delft, and he was the executor of Johann Vermeer, the painter. Vermeer died in his 40s, and he was the executor of his will. And there is a hypothesis that some of the early illustrations of Leeuwenhoek were actually drawn by Vermeer. There's an article from somebody from Japan. And what was unusual about Vermeer is he did two paintings of a scientist, Nobody else would do such a thing in the 17th century. And this is one of them. It's a picture of a scientist looking at a globe in a blue robe. And the one painting that we have of Leeuwenhoek has that globe and the same coat in it. So it's possible that this is from the Louvre. It's possible that we're looking at an idealized version of Leeuwenhoek. I don't know if that's true or not, but it's such a nice story, I'm going to stick with it. Now, this takes us to 1700. The next 100 years saw improvements in microscopic technique, histology, and so on, and gradually cells were found everywhere, initially blood cells in animals, then subsequently in animals which are more difficult to look at. And it was being proposed by the natural philosophers in Germany that you should consider living organisms, animals for example, as assemblages of animalcules, which is what the microbes were called at the time. Assemblages, in other words, of cells. And by the first part of the 19th century, the cell theory or cell doctrine was born by Schleiden and Schwann. This is a rather serious German scientist, Theodor Schwann, who in 1839 published in German, this is a translation, we have seen that all organisms are composed of essentially like parts, namely of cells. So what this is of course a statement that the cell is the basic structural unit of life. That was followed about 20 years later by Rudolf Virchow, the founder really of medical pathology in actual fact, also German physician and Minister of Education, if I've remembered correctly, in Germany. And he stated in 1858, the second part of the cell theory, every animal appears as a sum of vital units, each of which bears in itself the complete characteristics of life. And what that is, of course, emphasizing is that the cell is the basic functional unit of life, the simplest entity that exhibits the properties of life. Now, this is a major milestone in biology. We don't recognize it as such, in my view, sufficiently, and a couple of the other ideas I think we do. But this is a major idea in biology. It underpins many other things in biology. Here is a modern picture of the cell, and it underpins, knowing about cells, all reproduction and development. If we look at a mammalian embryo here, you'll see those little dots if you're near the front. Each of those is a single cell. And that is, of course, formed from a fertilized egg. And here we see the egg being fertilized by sperm. And if I've failed to convince you that cells are not important and interesting, I will say one more thing, which is I remind that all of you once looked like that. All of us were once a single cell, and that's what we looked like. And if I fail to interest you, well, you've seen the exit. You know how to get out in case of a fire, and I recommend that's probably the best way to go, because it goes downhill after this, okay? So, very important discovery, the basic unit of life, underpins all reproduction and development, because you get repeated cell division to make an embryo, and still a current topic of interest. You barely, every week, pick up the newspaper, there is something about stem cells. Overblown, I know, but still out there as something that's really interesting, really important. And so that's my first great idea, the cell. Second idea is the gene. The gene as the basis of heredity. Now, from ancient times, I'm talking the time of classical Greece, it was observed that particular characteristics of an organism are passed down from generation to generation, from parents to offspring. And so scientists, natural philosophers, wondered, of course, about what the basis of that heredity might be. And this was a question that was tackled rather seriously by plant breeders in the 19th century who were concerned with improving crop plants, but also had, of course, an interest in what the nature of heredity might be. But they weren't making very much progress. There was a significant amount of work, But what was happening was obviously complicated. They would take one plant with a certain, same species, but a certain set of characteristics, cross it to another. The characters sometimes became blended, sometimes one would get lost, then it would re-emerge again, and really they didn't make much sense of it. And this was a problem that was essentially solved by Gregor Mendel, the great gardening monk, as I think of him, who was the abbot of a monastery in Brno, in the Czech Republic. I'm a trustee of the foundation now that preserves his artifacts there. And it was a strange times, really, because it was really an order of scientific monks. I mean, it's something that we don't quite have a sort of concept of today, but that particular monastery specialized in science. And Mendel, who came from quite a poor family, was trained as a physicist with the intention of sending him out to teach in that area. But he didn't do very well at exams, and he didn't pass at a high enough level for his physics exams. And so he didn't actually end up doing that. But he was very interested in science. He did a map of the moon. I've seen his little telescope that he used to look at the moon. He was a meteorologist. He would study how barometric pressure changed as, for example, a cyclone went through and so on. And he turned his mind to plant breeding. And it was very interesting what he actually did, because he argued. He did a number of trial crosses. Actually, he experimented with about half a dozen different species. And that's really quite important, because he discarded 5 of them and decided to focus, in fact, only on the pea. I'll show you that in a moment. And the reason he did that was because he couldn't make much sense of the other 5 species he was studying. And there were reasons for that, which I'll explain in a moment. But there's a lesson for that about science, that you do try to be objective, but just sometimes you can't understand everything. You know, you can't do it. And there is a knack in choosing material that you can understand and you can make progress with, which is what, in fact, he could do. So he had a research program. I like to think about it. This was his garden. I took this picture when I visited there in 1981 in the middle of the Cold War. A very large garden, actually, and he had a greenhouse. It's rather like having the monastery was like a research council today. He'd built a big piece of infrastructure, a glass house of the same size. So he was well supported, in actual fact, well supported as the monk for doing this. Anyway, he decided to work on the pea, and partly because it had very clear distinguishing features within the same species, like tall plants, short plants, different coloured petals, different coloured seeds, and so on, so that it was quite simple to look at the different characters. And he argued that to sort out what was happening with these complex crosses with characters blending and re-emerging, you had to do several things that naturalists, who were mainly doing this work, really didn't do very much. You had to, accurately catalogue the types of plant forms that you were seeing. So you couldn't be just qualitative about it. You had to accurately have classification of what you were seeing. And secondly, you counted what you saw. So you didn't just describe it, you counted what you saw. And that made a big difference. Probably came from his physics background, because it's more quantitative. And for example, the very famous 3-to-1 ratio, which I briefly mention in a moment, was also observed by Charles Darwin and published by Charles Darwin, and he simply described it and then moved on. He didn't sort of try to do anything with it. Anyway, 3 reasons I think for his success. One I've mentioned, he chose material that he could make sense of. The second, he recorded accurately and counted, as I've already said. And the third one is he generated a theory, and then he went on to test it. Again, not so common yet in biology, which was still primarily observational. And what he observed was that in the first generation after a cross, often, but not always, one character would dominate from the 2 parents. But that other character re-emerged in the second generation when they were crossed one with each other. And he then counted them and saw, for example, that when you did that, and many of you will be familiar with this from sort of school biology, he observed this famous 3-to-1 ratio, which, as I said, Charles Darwin also observed in snapdragons in his particular case. And seeing those ratios made him think that maybe this was the sorting out of combinations of particles. That's how he sort of thought about it, because it was sort of combinatorial. So it led him, and this is the typical example: a green seed with a yellow one. You get yellow, and then you get 3 yellows and 1 green, and so on. And it led him to propose what is essentially a particulate theory of heredity. That's the way really to think about it. That there's particles, we'd now call them genes, and that these particles influenced or determined the characters. I have to be careful with the language because they don't quite determine the characters, but it's a shorthand. That secondly, that in any particular plant there were 2 particles that were important for determining that character, but only one of those particles was passed on in each gamete when you made the pollen or the ovules. But on fertilization, after pollination, you got one particle from one and one particle from the other parent, and then you reform 2 particles in the new plant. And that one particle might dominate over the other in determining what the character was. That was the sort of logic that he went through. Implicit in that, but not actually mentioned specifically by him, is the idea of information, that the particles actually transmitted information. And he didn't state that very clearly, but that is implicit in what he said. Now, this was a brilliant piece of abstract reasoning, quite frankly. Here he was, sat in his monastery doing this, and he came up with this brilliant piece of reasoning, absolutely right, but nobody took any notice of it at the time. And it wasn't that his paper was not read. He published it in a reasonably good journal, with a high impact factor, I'm sure. Joke, yes. And I even found by accident a reference to it in the 8th edition of the Encyclopaedia Britannica, which discussed what he'd done, but took no notice of his interpretation. And then it was rediscovered 35 years later. This was 1865, rediscovered at the turn of the 20th century. And it was discovered within a year by 3 groups, essentially working independently, who came up with the same ratios and the same sorts of conclusions. The first of them, and I'm not going to tell you who it was, tried to publish without referring to Mendel at all. The second one, who felt, I suspect, that they'd been scooped by the first, then thought it better to refer to Mendel so that nobody would have priority. You know, scientists don't change. We're all ghastly, of course. And what happens is that in the end, it all came out that they had been anticipated by Mendel, and he'd got it completely right. Why was that the case? Well, I don't think anybody really knows. I'm going to offer you a possible explanation, because between 1865 and 1900, there had been advances in cytology, and this is an example of a picture of an onion root tip, and there you can see in there, I hope, it doesn't, look, you can see it here. These are the chromosomes. And what people had seen in cell division was that every time a cell divided, you saw these chromosomes appearing, and these chromosomes would separate. And that brought to the idea that maybe the chromosomes were determinants of heredity, and that these were being divided into each cell. And then somebody called Van Bedderden was looking at a nematode, and this particular nematode only had 4 chromosomes, so easily countable. And he looked at the gametes from the nematode, and there were just 2 chromosomes. So now you see that reductional division that Mendel had postulated. He didn't call it that, of course, but the reduction from 2 to 1 was actually being seen here in the observations that were being made under the microscope. So I wonder whether Mendel's reasoning was so abstract that it didn't convince, and you needed a concrete sort of reality to connect that with the abstract reasoning. I don't know what was going on in their heads at the time, but I wonder whether the connection with the reality, concrete reality, made it easier to digest. Well, this was a great idea, and in many ways the 20th century was the century in biology that saw the development of genetics. We saw that the fact that DNA, deoxyribonucleic acid, was the hereditary material. That was done in the 1940s by extracting DNA from a bacterium, pneumococcus, and showing that the traits of that particular type of bacterium, strain of bacteria, could be transferred to another pneumococcus species, and that was therefore, they suggested that DNA encoded the information. This was done at Rockefeller University, which I was president of until a couple of years ago. A great discovery in the 1940s, made by Avery, McLeod and McCarty. And when I went there in 2002, 2003, this little old man came up to me and introduced himself to me at the sort of cocktail party they had welcoming me, and he said his name was Mac McCarty, and I sort of looked at him and I said, "You're not the Mac McCarty," stupid thing to say, I realised. And he said, "Yes, I am the Mac McCarty," and of course I'd been taught about him in 1960s, and I thought he'd been dead for sort of decades, and here he was, still coming to talk seminars and so on in his 90s. I mean, amazing, amazing man. That set the stage for, Crick, Watson, Franklin, Wilkins, and so on. The structure of DNA, I'll say something a bit more about that later. The double helix came out in the 1950s, and the central dogma, as Francis Crick called it, where the DNA is transcribed into RNA, nucleotides, determining nucleotides, and then translated into the amino acid sequence of the protein, which is the basis of how proteins function. And I'll touch upon that a little bit later. So this is indeed a great idea because it encodes the notion hereditary is particulate. It's based on DNA, and there's information encoded there. So the gene, my second idea, basis of heredity. It can give an explanation of genotype, the composition of genes, to phenotype, that is how we are made up, and has clear implications for what we are. How much are we determined by our genes versus our environment? Yet again, something that is always out there in discussion, something we still wrestle with. Very important idea. Origins in the 19th century, but something that we still really is current in all our thinking. Our third idea, evolution by natural selection. Now, you could be forgiven if you go into your bookshop, local bookshop, and look at the popular science shelves of thinking there are only two ideas in science. One of them is string theory, and the other is evolution by natural selection, which seem to dominate much of what you read. But this is indeed a great idea, and is probably, at least in my view, a very powerful and beautiful idea, probably the most important idea in biology. It's certainly central to biology. Now, the idea has two parts. The first is that life evolves, that is, there's a tree of life. That's the evolution part. And the second is that a major mechanism for evolution is natural selection. The two parts, and I make that distinction because Charles Darwin contributed to both of those, but particularly to the second. Now, Charles Darwin is the man over there on the right. This is him in his 50s. But I want to say a bit before we get on to Charles about his grandfather, Erasmus Darwin, who is the man there on the left, who's clearly a Georgian. I mean, clearly looks as if he's from the 18th century. And I mention him because a number of naturalists and scientists, actually particularly in France, like Lamarck, had talked about evolution prior to Charles Darwin. And Erasmus is typical of it, of one of these people, and I'm going to say a few words about him because he's rather more interesting to talk about, to be quite honest, than Charles Darwin. He was a doctor in Lichfield and in Derby, and he was a member of that wonderful society called the Lunar Society that many of you may have read about, which is a collection of scientists, entrepreneurs, social reformers, who met once a month under the full moon, which is why they were called the Lunar Society. Some people called them the lunatics, I believe, as a consequence of that. And they would come and talk about all sorts of things over a dinner, And if you go to Soho House in Birmingham, you can still see the place, one of the places where they met. They then rode home afterwards, I suspect, under the full moon. That was why they met, under the full moon, so they could see the way home. I suspect they were a bit drunk as well at the time, but that's something else. He was a very fat, charming man. He cut an oval out of his dining table so he could sit closer to his dinner. He had a terrible stutter, but was a great conversationalist. I mean, one of these sort of bizarre characters. He was famous as a doctor. George III wanted him to be his physician. He refused that. He was a sort of a bit of a Republican. He was famous because, like most doctors, he didn't actually cure anybody very much, but he was very good at telling you if you were likely to die or not. And this, I believe, was probably very helpful in setting your, family affairs in order and doing those difficult things if you needed to do them towards the end of your life. He was a scientist. He was very famous as a poet. He was a doctor. And he wrote all of his science up in verse. It's in these great poems, and I have a number of his original books. They're just amazing. He was a proponent of female education. He had 2 illegitimate daughters, and he set them up to teach a public school for girls, the first public school for girls, I believe, in the UK, and was quite a radical. So he's interesting person. There's a museum in Lichfield if you're interested in Erasmus, and 2 statues actually. Anyway, he argued that life evolved. And his motto on his coach was everything from shells. He had that. It was in Latin, of course. Omnis, conscious, omnis a, something of that sort. Anyway, and he argued everything from shells because shells, mollusks, of course, are formless and not sort of very interesting. And he was arguing that all complex life forms could come from these very simple structures, the mollusks. But, and he lived in the Cathedral Close in Lichfield, the dean of the cathedral did not like this, because it obviously denied creationism, and told all his rich patients from whom he obtained his income that they shouldn't go to him because he was a heretic, and so he had to paint out his motto, which he did, in fact. Anyway, he had the idea of evolution, and some say even some sense of a little bit about natural selection, but he didn't really say that very clearly. But it was his grandson, Charles, here on the right, who gathered, and this is of course crucial for science, significant, vast even, amounts of information, if you read The Origin of the Species, supporting the idea that evolution had taken place based on both the fossil record and on his analysis of living animals and living plants. And just as an aside, the importance of evidence to support a scientific idea is really important. So the 18th century speculators, important as they were, they did not have evidence to base their speculations on in the same way as Charles did, which he gathered on his 5-year Beagle trip, that many, that we all sort of really know about. Then he retreated to Down in Kent, not writing The Origin of Species until over 20 years later, till 1859. And in this book, there's one illustration only, and it's this, where he built the first tree of life. And you'll see this tree of life here, where living species are at the top. There's a tree, everything is related, or nearly everything. There's lots of stuff from A. He sort of kept his options open as to whether, you see, he didn't, notice it's not rooted. Under A to L, it's not rooted. So he probably wasn't quite clear whether life had arisen more than once, or whether he was a sort of deist at the time, and whether it had been created, the initial life form, it just wasn't clear. He later became an agnostic, but at the time, I think he was probably a deist. So this, of course, tells us, it's the first real demonstration that all life is related by descent. In other words, every living thing is connected by descent. And what he did is he observed that living organisms were often very well adapted to their lifestyles and their environment. And he had a number of instances of that. The famous one are Darwin's finches from the Galapagos, where the beaks are adapted for particular functions, to get insects, to break nuts, or whatever. And I found this picture here. It's just a nice one. We have Darwin's finches on the left, and we have all these pliers on the right, all of which have been designed, in this case, intelligently designed on the right, to do different things. And just like it looked as if the beaks had been intelligently designed on the left with natural theology, but which Darwin came up with an idea of evolution by natural selection. We usually associate evolution by natural selection solely with Charles Darwin. He was not, however, I should say, the first to propose it. It was proposed in a book published in 1831 by Patrick Matthew, who wrote a book on naval timber and had a two-side summary of evolution by natural selection, extraordinary, which nobody took any notice of. And of course, as is much better known, is Alfred Wallace sent this famous letter from Borneo explaining the same idea, and they both co-published in 1858. But it is indeed Charles who put together the thinking and all the evidence in a much better way. So what is natural selection? The idea is, just briefly, is that within a species there are variants. And these variants are due to inheritable differences, we now would say genes. Those variants that are most successful reproduce more because they grow better, they grow faster, they get mates, and so pass on more of their genes to the next generation. So-called survival of the fittest, although that isn't a term that Darwin used. Spencer was the first to use it some years later. This biased handing on of genes gradually led to changes in the characteristics of the species, that is evolution, and eventually that can be sufficient to give you a arisal of a new species. So that's a sort of a summary of it. Now, I want to repeat that argument, but I want to link it, in the way I repeat it, to the two earlier ideas that I've talked about, the cell and the gene, because these three ideas are in fact related. And this was an argument that was first made by the geneticist Muller, who discovered mutagenesis, essentially. And what he said is that living things have properties which allow them to undergo natural selection and therefore to evolve. And those properties are the following. Life must reproduce, obviously. Secondly, life must have a hereditary system whereby information defining the characteristics of an organism is inherited during that reproductive process. That's the genes. And thirdly, the hereditary system needs to exhibit variability upon which selection can work. And if a living thing has those three characteristics, it will evolve. And those characteristics are implicit, of course, in the cell and genes within a cell. Because if we look at a cell, and we have one here which has got a yellow coat, and then at the bottom a gene mutation gives rise to a red coat, cells reproduce, that's the first thing. They have genes that define the characteristics of the cell, and in this particular case, those genes, of course, are in DNA. It doesn't have to be the only chemical that might be hereditary, but it is in life forms as we know it. That the chromosomes replicate every division, you get heredity, but mistakes can occur or mutagenesis can occur, and on the right-hand arm, the gene that is important for being yellow is changed to a gene that is red, and that is the variability, that genes exhibit variability, and once that's copied, it's maintained. And if red cells are at an advantage over yellow cells, bingo, you've got evolution by natural selection. But the point about it is it comes out of the idea of the gene and the idea of the cell. You just have to introduce imperfection into it, that is variability in heredity, and then that will actually come out. So, this is a very important idea. It was, as I said, mainly Charles Darwin. He ends The Origin of the Species. I just want to say it's such a beautiful sentence. The last sentences of the 1859 book. Whilst this planet has gone cycling on according to the fixed law of gravity, referring to Newton, he wanted this to be a law. You know, that was why he wrote that. From so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. So this linkage to a law and a statement of the creation of life. So, evolution by natural selection. What are the components of the idea? All life evolves and is related by descent. Very critical. 2, natural selection, the major mechanism. There are other mechanisms. Darwin himself thought there were other mechanisms. And there's other things that make this also very, very current. Kin selection, the idea that if you sacrifice yourself for your 3 brothers or 3 sisters, then you will be passing on more genes to the next generation, because they will have enough of your genes to make it worthwhile you looking after your brother and sister. This is work of Bill Hamilton and others, is important to understanding altruism and even perhaps morality. I mean, we have to be aware of that when we're thinking about these issues in moral philosophy, which normally we think quite separate from biology. And the last point, which I really liked, which I first heard from Guido Pontecorvo, who worked at the Imperial Cancer Research Fund, was that if we recognise that we are related to every living thing on the planet, do we not have a responsibility to be stewards for all our relatives? It's a sort of statement about protecting the planet, protecting the biosphere on the planet. So this is an extremely powerful idea, this third idea, but the definition of life that emerges from it is essentially historical. It tells you where life comes from, but it is less satisfactory, really, in telling you how life works. It doesn't sort of give you a sense of that. It tells you how it gives diversity, but not how it works. And for that, we move to the 4th idea, and that 4th idea is life as chemistry. And this is a mechanistic basis for life, chemistry and physics, and I'm going to focus on chemistry. Now, the reason it's worth mentioning this, a mechanistic basis in this way, is that for many years, from Aristotle onwards, it was argued that living things had a natural end or goal, a movement towards a goal, which of course they do, it's a teleology, and that was driven by an inner vital force different from the laws of physics and chemistry, I mean, which was the origin of what's called vitalism, to contrast it with mechanistic explanations. And vitalism got in the way, really, because it's like the ghost in the machine, which is Koestler's book about motion, but it's, of course, becomes sort of unknowable if it's something that's very special. But it did dominate. And the replacement of that idea that biology could be understood in terms of chemistry and physics was really important. And that occurred, started to occur again in the 18th century. I mean, everything I'm telling you about, we're not talking, 20 years ago. We are talking 300 years ago in some of these cases. And the first is Lavoisier, who was important here. Now, we think of Lavoisier as a chemist. This is a wonderful painting from David, now in the Metropolitan Museum in New York. It used to hang in the president's office until they had one of their financial crises and had to sell it to pay off, the debts. And there he is with his beautiful assistant, who happened to be his wife, and also his laboratory assistant there. And he has the most wonderful leg, don't you think there? I think it's a great portrait. Anyway, what Lavoisier did, apart from founding modern chemistry, is he studied fermentation. Being French, of course, he was interested in making wine, and he was interested in how grapes made alcohol. He measured the sugar level in a grape extract and showed it was turned into ethanol, and you only needed small amounts of biological material, we now know to be yeast, which seemed to be irrelevant. So he began to think that there were changes that were catalyzed by the biological material, grape or yeast or whatever was there, that turned one chemical into another. Unfortunately, his research was terminated by the revolution, because and you can see another picture of Lavoisier, and you see the guillotine there on the right this time, because he was a tax collector. And although he was a supporter of the revolution, when it went all crazy, in 1794, he lost his head. And he was judged, and the judge said about him, when it was argued he was a supporter of the revolution and he had great things to contribute as a scientist and he'd done all these things, the judge said, "The Republic has no need of chemists and savants." I always think of that when we have to toady with the politicians, you know. They'll have our heads if we're not careful. So, it stopped for a while, and then Pasteur. So, this is clearly Francophile. And Louis Pasteur, 1850s, demonstrated that the chemical transformations that Lavoisier began to talk about in fermentation was due to living microbes. And he was doing an applied project, and I think this is always very important when people start making the distinction between basic research and applied research as if they're very different. He was doing an applied project in Lille on sugar beet fermentations for making sugar, which the manufacturers found often went sour. They'd make acetic acid, instead of sugar. Making lactic acid, instead of alcohol. And he looked at the material under the microscope and saw when the fermentations worked, they had yeast in there. When it didn't work, they had bacteria in there. So he concluded that the different microbes were responsible for the chemical transformations. And so he could tell the Lille sugar beet fermenters, just take some yeast and put it in there and it will always work. So they paid him, right? But then he thought, what does this mean? And he concluded that fermentation, I'm quoting this, in French originally, fermentation was a physiological act yielding chemical products necessary for the cell. And such chemical reactions are an expression of the life of the cell. Absolutely laid out. Absolutely beautifully laid out. And came from an applied project, but he applied it to a basic understanding. And it was on that in which, of course, the science of biochemistry was built, where you explain the living processes in terms of chemistry. And that is based upon, not completely, but to a great extent on proteins. Proteins are little chemical machines, they're catalysts, which act to change one chemical to another, so they break bonds and reform bonds. They are made up of, proteins are made up, enzymes as they are made up of a chain of amino acids, and the chain of amino acids determines the structure of the protein, whether it has bulky residues there, whether it's charged, whether it likes water or not. And what that makes is a molecule with particular surfaces that can undertake, under very mild conditions, certain chemical transformations. That's essentially what's going on in a particular enzyme. And what these are doing is creating little chemical microenvironments within the environment of the protein. But it is extraordinary that within a cell, many hundreds, if not thousands, of those chemical reactions are going on simultaneously in a few cubic microns. Extraordinary. This is a metabolic chart, and each of those dots in central metabolism, the red circle there is the Krebs cycle, represents a chemical reaction. This is only the central part of intermediary metabolism. It doesn't touch everything else that's going on in the cell. And the thing I want to emphasize here is all this chemistry is going on simultaneously in a cell. If we try to do it, or at least conventionally in chemical industry, we'd have some big reactor doing one or two of these reactions. And this is wonderful. This is just amazing what they're actually doing. And the way I like to think about it is that this is the basis, really, of structure in cells. The way I like to think about cells, where you also have structure, you have different membrane-bounded organelles, you have complexes of enzymes, and the structure which we sort of look at the cell, which we often divorce from what it's doing, I like to think of as simply lots and lots of different compartments which allow chemical reactions to take place simultaneously in the same cell. It's a sort of a different way of thinking about it. So I think of the cell as a myriad of chemical microenvironments formed by enzymes, complexes, protein machines, organelles, carrying out all the chemical and mechanical work that's going on within the cell. And that is the idea of life as chemistry, provides a mechanistic explanation of life. it is, of course, the primary paradigm now for thinking about diseases, because we think of diseases when there's something that goes wrong in one of those chemical reactions, or a number of those chemical reactions, and that provides explanation for diseases. And that thinking is central to much of medicine and the pharmaceutical industry in trying to produce drugs or interventions that can alter or compensate for those particular chemical defects. So vitalism has been killed by this, and it's increasingly important for treating disease, and is the basis of much translational biology. So that's my fourth idea, and if you're still up for it, I can give you a fifth idea. Take a deep breath, we're getting near the end, and you only have to concentrate a little while longer. Now, my fifth idea, which is one, is absolutely not my idea, in fact, once again, it's an old idea, is the idea that biology is explicable in terms of the management of information and dealing with a complex system. And this is an idea that's been talked about a lot, but it's not one which is new. Thinking about biology as complex systems has been around for many years, and I actually discovered when I looked into this that Kant, the philosopher, at the turn of 1800, actually wrote about 30 pages on biological organisms as complex systems. I mean, in a book on moral philosophy, goodness knows why he went into that in moral philosophy, but he really, really did. But the focus on information, which has broken this area open, really came into place in halfway through the last century, particularly at the beginning of computing, where there was a big thinking about information and information management in computing, and this played a big role in theoretical thinking about biology. And some of you may remember Conrad Howell Waddington, who was in Edinburgh I met him in the 70s here, and he thought a lot about information, as did Henry Catcher, which some of you may know as well. Anyway, that was thought a lot about in the 50s and 60s, got somewhat eclipsed by the detailed of molecular biology, which focused on single components within the systems, never really completely eclipsed, but has come back with a vengeance in recent years, although it's now thought of being a brand new discovery, when of course it absolutely, of course, is not. Now, the point about this is, and the way I like to think about it, is that many biological phenomena can be described in terms of interacting components that make up a complex system, that's one part of it, and can be understood best if you think about them in terms of the information flow through that complex system. So it's a combination of both a complex system of different components and information flow through that system. And these systems in biology operate at different levels. The one I'm going to focus on is chemical systems for cells. Right? But you can also think about it in similar terms for neuron systems in brains, for example, or ecological processes in ecosystems. I mean, it is sort of scale-free. The sort of thinking occupies different levels. But I'm going to focus on cells because it's the basic unit of life, and I think it illuminates something about life. So the idea is to explain higher-order biological phenomena, phenomena like homeostasis, communication, reproduction, organization in space, you know why we have our shapes, organization in time, all in terms of information and information management and the systems that do this. And this approach emphasizes regulation, network, organization, emergent behaviors, whatever that quite means. You have to follow the information. And I've written this down so I get it right. In short, what I'm saying is that you need to translate the descriptions of the chemical and physical processes being carried out in cells into the underlying system and the management of information within that system. And by management of information, I mean gathering information, storing information, processing information, and using that information to determine the outputs of the complex system. And that is an objective on top of describing the chemistry. Now, to illustrate the point I'm trying to make, I'm going to give you two extremely well-known examples, but emphasize them in the terms of what I've just said. The first is something I've mentioned already, which is the structure of DNA. Now, the double helix is a beautiful structure, but it is not beautiful really because it's a structure. It's beautiful because it is a beautiful idea. And the idea that is in the structure of DNA is that it is essentially a digital information storage device. That's actually what DNA is. Digital information is encoded in the order and type of nucleotides that make up each strand of the DNA. And the chemical structure of DNA actually underpins heredity, but you only understand it when you think about it as encoding information. If you only think about it in terms of angstroms and what atoms are touching other atoms, you don't understand it. You understand it, as Crick and Watson so clearly saw, you understand it when you translate that into the storage of information. And as it turns out, the ready copying of information every time that molecule is actually replicated. So it's only when the chemical structure is described in terms of information that it makes biological sense. And that's the point. You can describe it chemically and it will make no sense at all, but if you describe it in terms of information, it makes biological sense. So that's one example. Second example from a similar time period, really. And to introduce this, I'm going to show you a picture of what is called a governor. I took this picture in a steam boat in New Zealand, actually, in a boat made about 1890. And I don't know if you know how you can understand machines when they were built before 1900 and virtually no machine when they're built after 1900. So you're forced, if you're a biologist anyway, to go back to things pre-1900. Now, what this is happening here is that this spindle is rotating. The balls come out due to centrifugal force, lifts up a valve, cuts off steam to the engine, slows the engine down. It's a negative feedback loop, and like the thing that we have on the top there, but now we've translated it into a biological sequence of reactions, and this came from Jacob and Monod in the Lac Operon around 1950s, 1960. And what this says, and we see it in the negative feedback loop, just like the governor, A is a metabolite turned to B, turned to C, and C, when it accumulates, switches off the enzyme that switches A to B. So if you get too much C, you switch off the manufacture of C, and the thing is homeostatic. You can play around with such loops. A positive feedback loop is at the bottom there, which is actually turns the whole thing into a switch. As against homeostasis, you go from A to B to C, and C promotes the catalysis of A to B. So once you start getting a bit of C, you switch the whole pathway on. So that is a sort of a positive feedback. Now, playing around with such circuits, you can do all sorts of things. You can build up on this. I don't want to do that today. But you can get different feedback controls, you can make switches, as I've said, you can make timers, you can make oscillators, you can make toggles where you toggle from one behavior back to another. And if you combine all these things together, you can do lots of quite complicated things. And many of us who do sort of molecular and cellular biology are putting these control circuits together to try and make sense of what we're doing. But once again, it's the chemistry of the networks which we're trying to translate into information flow to understand them. And a good analogy here is an electronic circuit. I stole this from some journal where some wit had sort of replaced the transistors with different oncogenes here to try and emphasize that they're all connected in a regulatory circuit and make sense when you understand those connections. And it is those connections and the management of the information that's important. However, we shouldn't get carried away with this analogy, because biology is much more sophisticated. Dennis Bray has compared a cell to a computer like this, but it's a special sort of computer, because here everything is hardwired, so you do it all by the software. But in cells, he coined the word wetware. That is to say, you connect through diffusion within water, so it's wetware. And what that allows you to do is to rewire the network. So not only can you play with the software, which say in crude terms you could imagine the DNA plays a role as software, but you can also rewire the hardware by reconnecting the molecules in different sorts of ways. So that makes it more complex and more versatile than a computer. So you need to consider the cell as a complex chemical system, wetware, which acts, and again these are the words I want to sort of drive home, it's a sort of logical computational machine. It links logical modules of the sort I've said to manage information. Now I'm gradually getting more and more sort of out of it at the moment, okay, so just follow me for a bit longer, because now I'm just going to say, where could this lead us? And there's various analogies and metaphors that are interesting. One is that complex networks we come across in different levels. Here is an aeroplane route in the United States, okay? And you've got a hub-based system, you know, where you always have to fly into New York or Chicago or Cincinnati or somewhere to actually get to somewhere else. And what this tells us is if you take out New York or Houston, you're in big trouble. But nobody really cares about Kansas City at all. And the people study these networks, and the connectivity of them is quite interesting because it tells you something about how the complex system works, and there are more analogies than you might imagine between these human-made networks and the networks that exist in biology. Another one that is worth thinking about, that is that when we think of networks and pathways, our brain is wired to, as far as possible, to get systems organized in simple, linear, causal relationships. It's just, you know, it's just how we work best. So we end up with that linear pathway at the top, and you can easily understand it. If you knock out the first arrow, the last arrow doesn't happen, okay? But the reality in biology is something more like this stuff at the bottom, you know, where intuitively, if you take out this, you can't work out quite what's going to happen. You know, it's not intuitive. So biology is complex. It's redundant. I'll come to that in a little moment. And so it's difficult to actually work out and understand. And the way I sort of like to think about it is that we need to have a different sort of thinking. And the next slide I was always too frightened to show in the United States, but I think it'll be okay in Scotland, okay? Because the key issue is here. We think a bit too much like men at the top, you know, and they just have an on and off button, and women who are more sophisticated in this respect and more complex, and we need to think more like women. I got this out of a pilot's manual, actually. And I normally annoy somebody when I put that up, but I hope I won't today. Now, the next thing I'd like to talk about, just to get you to think about it. If we have a signalling pathway like this, then in its simplest form, you switch this on, you switch this on. But if you now introduce dynamics into this system, that is to say, if you switch on the first arrow for a period of time, then switch it off, okay? You can now use the same circuit to communicate quite different messages. Because if you look above, you'll see that we have two messages, one where in a unit of time you have 4 pulses, another where you have 2 pulses. So simple networks like this can be made much more complex if they're made dynamic. And this has quite significant consequences, and I can only talk about this in metaphor. If you have a simple system on-off, you'll have a traffic light system, you know, it's green or it's red, right? If you introduce dynamics, you can have the Morse code. And once you have the Morse code, you can write the works of Shakespeare. I mean, the amount of information and its management, once you introduce dynamics into a system, is much, much greater. And we now, gradually it's emerging, dynamics is really important in understanding signalling, and we have yet scratched the surface, in my view, and we don't yet understand that. So much to learn. Temporal and spatial organisation crucial. In phage, one of the simplest things with an actual structure, it's all direct molecular interaction, very obvious, ribosome would be another one. But if you want spatial order beyond direct molecular interactions, you actually have to have spatial order at a higher higher scale. And this is is Turing's famous equation, and this is a Sabatinski reaction, where a simple reaction can produce order over millimeters. And this is again only a metaphor, that we have to think about how you can get spatial order beyond direct molecular, direct atomic contact, and that will require different ways of thinking about it. Now, we have to remember, a final point I would really want to emphasize here, in thinking about these complex systems, is that they are not intelligently designed. I say it again, they are not intelligently designed. Harrison's clocks are intelligently designed. Harrison was the guy in the 18th century who made the chronometer that would allow you to determine longitude. Darva Sabel wrote a nice book about it. You can see the 4 clocks in Greenwich. How was this intelligently designed? He did Harrison 1, top left. He studied it, he put it together, it didn't work properly. He took it all apart and he made a new one, completely new, top right, Harrison 2. Then he made another one, Harrison 3, and ended up with Harrison 4, which is absolutely nothing like Harrison 1 or 2 or 3. He was designing, learning and redesigning. We have evolved not by going back to the drawing board again. We have within us our ancestors, and we are tinkering with everything we had before. And what that means is that we will not be perfect machines. We will have all this redundant stuff, and all it is that we just make do. You know, we tend to think we're perfect and that cells are perfect. They're not. They just get by. And that means that the systems are not perfectly designed. They're often redundant. They're often extremely complicated. And to think of them as perfection is not actually the best way to do it. So where I'm getting to here is that we need to turn the biochemical descriptions into information management, and we have to understand that this will be quite complex. And my final point is this, that the explanations that may emerge from the complexity may well be counterintuitive. This is the final point I just want to explain. Because us biologists, on the whole, work and live in a common sense world. When we're doing molecular biology, cell biology, physiology, ecology, or whatever, we make pictures of what we're doing in our heads. I'm a cell biologist. I imagine this room as the cell, and this is touching this, and it's all common sense world. Now, this emphasis on complexity may take us into a stranger world because it isn't so intuitive. And physics did this 100 years ago. Because when physics got very big and very small, you know, with the universe and with the atom, things got increasingly bizarre. Physics with Newton is sort of common sense, you know, more or less. But things got strange. If you take Einstein, relativity, 1905, it gets a bit tricky. I'm sure you're like me. You'll buy a book that explains relativity. You read it. You sort of get it, you close it, and it slips away, you know? It just slips away. And it slips away because it doesn't quite make sense in the common sense world, you know? And you have to hold it in a sort of different sort of way of thinking. And if you think you can cope with Einstein, move on 20 years to quantum physics, and that's utterly impossible. You know, you have dead cats and live cats at the same time. It is very, very strange. And it only makes sense, really, within the mathematics of what you're doing, and it isn't part of the common sense world. I learned this, my daughter works at CERN, she's a high-energy physicist, and I realised she doesn't understand it either, what she's doing. But they know how to manipulate it and how to sort of deal with it, but it doesn't make sense in a common sense world. And what I wonder is whether the complexity of biology may take us into a equally non-common sense sort of world where we need help from the physicists and the mathematicians and those who are better used to thinking in an abstract way to actually try and make some sense of it. I don't know if that will be the case, but I think it's possible. Well, I've talked about 4 ideas that I think most people can agree with. They're not the only ideas, of course. The cell, the gene, evolution by natural selection, life as chemistry, an explanation of living systems, but I also think information and its management and complex systems will be very illuminating about biology and explanations for biological phenomena in the future. Thank you very much. Thank you.
Well, ladies and gentlemen, 4.5 centuries of discovery in biology in just over 1 hour. And the last 2 minutes, he said, we're not going to understand it anyway. So now is your opportunity to ask questions. You have to remember that there are 2 male buttons here, simple on and off buttons. So please keep the questions simple. Thank you. And short. Who would like to ask the first question?
And I'm a bit deaf, so do shout, please. And I do like questions, by the way.
Microphone coming.
Microphone, wait for the
so
mic.
everybody
It's
else
coming.
can hear.
So obviously it is, all these things are, concepts are incredibly complicated. The one thing that is still lacking in biological knowledge is thermodynamics and physics and how the chemistry links to energetics really. And when do you think that's going to come, and what? Because at the moment that type of research is not funded very much, is it? Really?
No, obviously we have to accommodate thermodynamics. Obviously, biological systems work in a strange thermodynamic domain. I think that does emerge from the fact that we're dealing with structures that are in a sense purposeful. Mono, you invented the word teleonomic as against teleological in purpose, but not due to vitalism. I do think we need to explain biological phenomena in terms of the physics and chemistry, as you say. We need to do it, and dissipative structures, which is where you're going. But it may be that explanation won't come so much in focusing on the chemistry so much as on in these higher order things like information, where I think we can make more rapid sense of it. So I think we always have to have our eye on that stuff, but think about it in the sort of biological context.
I also think quantification is really important, i.e. how you relate. You've got networks, but actually the levels of things and how the levels of things relate to function.
Well, isn't it interesting, I emphasised how Mendel quantitated, and the naturalists didn't quantitate so much up until that time. And as I'm sometimes known as a molecular biologist, and we just sort of have a band and it's a bit more intense than another band, and that was enough. They won't let us get away with that anymore. We have to quantitate. We have to do it properly, though, and I have to say that that isn't always happening. We get spurious statistics being asked by editors and reviewers who don't understand statistics. So I think we've got to up our game a bit so that we actually do ask sensible questions about quantitation rather than just pressing the button that says on your computer t-test and then, quote it, which is often what happens.
Personally, I love the 5th big idea because it is all about what we have to do next. But I have one concern, and that's because the first 4 we take as established, understood, unequivocal, but at least a substantial minority accept the first, the second, the 4th, but not the 3rd. And does the 5th let them off the hook?
I don't think the 5th lets them off the hook. I think here in our society, we're okay with the 3rd idea, basically. Yeah. But that isn't the case, as you rightly say. And it's in all parts of the world. It wasn't the case in the middle of America where I was working. It was not a problem on the East Coast where I was, but it certainly was in the middle. Mm-hmm. And it isn't the case in many parts of the Middle East, for example, too. It's where we have this conflict between ideologies or religious thinking, where certain ideas are taught or accepted based on different sorts of criteria from science, where you believe certain things due to testimony, faith, reference to particular books and teaching. Whereas science, of course, approaches in a different sort of way. It's also not entirely objective, we have to be completely clear, but it is driven by evidence. Built into science is a skeptical approach to what you're doing, so that you attack your ideas in a way that you are unlikely to do if you were a political Marxist or a fundamental Christian. You would not want to attack your ideas in the same way as we should do. I'm not saying we always do, but we should be attacking our own ideas. And we will change our ideas if the evidence makes us do so. And that's what is so wonderful about the self-corrective nature of science when properly applied. And of course it goes wrong, but when properly applied. What I find interesting about it is you're talking about evolution by natural selection, but gradually I think science is going to impinge upon other areas that traditionally have not been the domain of science, but have been domain of the moral philosophers or the theologian, because we will find that we will learn things that are important, and touched upon it, altruism and making decisions, moral decisions, and at the moment we can't make too much progress there, but I suspect understanding how we think and neuroscience and so on will gradually inform such things. And just like in 1500, when theology provided explanations for the structure of the universe, which has now been abandoned by theology, or most theology, and is now clearly in what science does, I think we may see a gradual shift. So I think it won't stop at natural evolution by natural selection, but I think we just have to stick to the evidence and good thinking and keep calm.
It'll be all right in the end. Yes. Question at the back, yes?
Yes, like independence. Yes.
Yeah.
Oh, sorry.
Didn't hear that.
Talking about the natural selection.
Could you stand up so I can see you, please? Thank you.
Talking about the natural selections and how much now we understand it, and we progressed a great deal since we addressed all these things. Yeah. But we still haven't got a lot to say, I think, about why things go wrong as the genetic information gets stored and transformed. We address how it comes about, but when a mistake happens, we don't seem to address that.
Well, the truth is there's many things that we don't understand, many things, and you're quite right that when things go wrong and you get pathologies, often they are very difficult to understand. And I think and I'm glad you brought this up really, we have to approach these problems with a certain humility. I think we do have a basic paradigm within which we can operate, which is quite effective, but it is not yet at a state where we can explain many things, because there are many things we cannot explain. I chose to work on a really simple system, which is the cell and how it reproduces itself. I chose such a system partly so you could make progress with it. You know, the same decision that Mendel did to work on a plant. But I barely understand how a cell works after 40 years, you know, barely understand. And most of the world wouldn't care less about how a cell works, to be perfectly honest. And when we get to the sort of complex phenomena that you're talking about, we are really having to work hard. So I agree with you, but having said that, I am, as you've gathered, and you've already slightly made fun of me, an optimist, complete optimist, that gradually, over time, we will get to know more and more. I do think that. So I'm an essential optimist that we will make sense of it, but I'm an absolute realist that we have not yet done it.
Paul, towards the end of the talk, you mentioned the issue of redundancy, which, I mean, could you give us an idea of the scale of that and the extent to which, I mean, I raise it because of the last question, which things go wrong, but perhaps they go wrong because they have become too complex.
Or maybe the complexity, of course, some would argue, give you buffering to things going wrong. Do you know, one of the things that really has, I'm going to use the word shocked me. I work on yeast, fission yeast. There's 5,000 genes. We, over the last, we finished this project 2 years ago. We deleted every gene one at a time in that organism. Okay, it's been done before in budding yeast. We did it in my lab in fission yeast. So we had 5,000 strains, each of which had one gene deleted. Okay? Yeah. And if you now look, you find that 75 to 80% of those strains, it's actually 78%, it's perfectly happy under the conditions under which we're growing it. In other words, you can dispose of nearly 80% of the genes. And this slightly bothers me, okay? Because as you know, evolution by natural selection, you'd think everything is under selective pressure to be maintained there.
Yeah.
Now we have what I call the ecological cop-out, as I call it, which is that we have yet to devise conditions under which they are important. Okay, so only in the mollycoddled laboratory is that the case. But you know, I feel a certain discomfort about this, and I wonder whether there's something else going on. And it touches on redundancy because it suggests that all these genes here, and yet you can pull, you know, you can get rid of many of them, you know. And just to back that up, I've spent 35 since I was working in Edinburgh in 1973 onwards. I've sort of researched the network of genes that control the onset of DNA replication, the onset of mitosis, so two straightforward switches. And over time, colleagues and myself, we've devised a network of genes, maybe 25, 30 genes, that are key to that. And in the last year or two, I went back in a sort of reverse synthetic biology approach, for those familiar with this, and sort of ask the question, could we simplify the network and still get it to work? And I'm shocked to tell you that we have all of these genes which we thought were essential and crucial for the proper reproduction of a cell, and we can get rid of over 80% of them, and it still works perfectly well most of the time. In other words, I mean, we had to fiddle with things, we had to put things together in particular ways that we hadn't done before, not saying it was that straightforward, but all this stuff there is not essential, it's redundant in some way. And this is why I went back to the humility bit. You know, there is a lot we don't understand, and as I come near the end of my working life, I realise I'm basically undermining much of what I did up until now. So it's time to take up gardening, I would say.
Two more questions, please. One in the middle, yes.
Well, following on from what you just said, would you like to comment on what's currently being called epigenetics and non-coding bits of DNA and how they bear on the complexity you're talking about?
Yes. Okay, epigenetics. First of all, a term that was coined in Edinburgh, or certainly by Waddington, and the genetics department here for a while was called the Department of Epigenetics. Once again, one of these things that looks as if it's been invented in the last 5 years, but he first wrote about it in about 1942. What is it? It is, actually, it means different things to different people, so it's quite difficult to define, and people get very excited about the different definitions and fight each other over it a bit. But basically what it means is it's heritable differences that are not determined by the nucleotide sequence of the genetic code, and it is had a biochemical explanation in terms of the proteins that are associated with DNA and the modifications of those proteins that can persist from division to division to division. And that gives you heritability, probably over a more limited time scale compared with DNA, but which can persist from generation to generation. And this is interesting, not new, as I said, talked about a lot 50 years ago, but what is new is that there's now a biochemical explanation for it, which means you can now do proper experiments to look at it. And it does indeed colour how you think of it, because if you see something inherited, or if you see a trait persisting for 5-10 divisions, then you may not only think about it in terms of locking a control system in a particular part of control space, you can think of it in terms of modifications of these proteins making up DNA. So I'd see it as not a breaking down any sort of new ways of thinking that are sort of are that radical, but it is absolutely another thing that cells do, which we didn't realize how much they did, that we need to take account of. So really important. Perhaps not quite as important as the popular rightists say, but really important, and we need to think about it when we're explaining phenomena.
Who's going to ask the last question?
Ooh, that's always a killer to say that.
Could I just mischievously ask you the last question, which, of all these 5 great ideas, which is the greatest? You have half an hour.
I have half an hour. I do think evolution by natural selection is a beautiful, beautiful idea. It does give, it provides in some sense an explanation of how complexity can arise without a creationist agenda. I think it's one that is very rich in its implications. That's why you see so many books about it. Has implications now in, for example, computer programming and how you can design how things work by setting up selection systems. So I think that's a very rich idea. It's the one that I think I like best, and it is a pity that there are significant numbers in the world who want to reject it for reasons which are not very sound.
Thank you, Paul. I'm going to now call on Sir William Stewart to present the vote of thanks. Will.
Well, how do you actually follow all of that? What can you say about it? It's been a privilege, Paul, and it's been a pleasure to listen to you tonight. It wasn't a lecture, it wasn't a talk, it was an exposition, wasn't it? And as President of the Royal Society, as Nobel laureate, Paul's got a license, he's got a platform to say anything he likes, and he does. And you know, down there in London, the people in the office tell me, the great problem with Paul is you don't know what the hell's coming when he walks through the door of your room. And you've got to be prepared for anything. And we were prepared for virtually anything tonight. But what he wants to do is that he wants to engage, he wants to debate, he wants to interact, and he wants dialogue, and he wants confrontation. And that's one of the great things that Paul does in his talks and in his thoughts in general. His original lecture, Nobel lecture, summarised the very precise work that he did in discovering with colleagues the specific protein molecules, the checkpoints, which help control cell division for good or indeed for bad. And I said, well, that's complex enough. Thank God he's getting on to talk about great ideas in biology and the history of evolution of the cell and things like that. But in addition to that, I wondered, he didn't stick to that. Among the plethora of issues that he made, there were several that came out tonight that I thought about. First of all, he told us about the mind-boggling advances in molecular biology, in biology in general. But what he also emphasized, and we don't always appreciate it, was the importance of chemistry and physics and the other natural sciences in all of that, and the importance as President of the Royal Society he's got to make and should make these points. And secondly, he made the other point that scientific advances, real scientific advances, come from the very brightest, specialised people working together with each other, rubbing shoulders and interacting off each other. And in addition, using the new applied technologies which chemistry and physics bring to the scene at the present time. And that was simple enough. He then touched on the fact that the workings of even the simplest cell are infinitely more complex than we ever imagined. And I got it as far as the unit of life. The cell's a unit of life. What is life itself? I wasn't quite so clear about that. But after that, he went on to talk about management of information, etc., information technology. Where were we going in the future? And at the end of it all, I said, what am I going to say here? And then Paul summarised it up for me. He said, I don't understand it myself. So if he doesn't understand it, how can he expect somebody who's proposing a vote of thanks to understand it? So I'll stop at that stage and simply thank Paul for a fascinating lecture. Gives us lots of things to think about, and I'd ask you, the audience, to show your support and thanks in the usual way.
Thank you all for coming in such large numbers. It's been a great evening and safe journey home. Good night.